So Alzheimer's disease is a pan-ethnic genetic disease that's known to exist in two forms: early onset familial Alzheimer's disease and late onset Alzheimer's disease. It's characterized by beta amyloid plaque buildup and buildup of neurofibrillary tangles characterized by tau protein filaments in the brain. So the risk factors associated with Alzheimer's disease are essentially age and genetics. Specifically, the APOE gene is associated with late onset Alzheimer's disease, and the APP gene is associated with early onset familial Alzheimer's disease. The paper we're presenting is titled APOE for Markedly Exacerbates Tau-Mediated Neurodegeneration in Mouse Model of Tauopathy.
So some background. So APOE is a lipoprotein who mediates the lipid transport between cells. So it's primarily expressed by astrocytes and microglia in the brain, but in certain circumstances also by neurons. So there's three different APOE alleles. So there's E2, E3, and E4. So E4 is known to have or be the strongest genetic risk for Alzheimer's disease because it increases the risk by threefold, and carriers are 8 times more likely to develop AD. So in addition to the APOE, there's also the tau-mediated Alzheimer's mechanism or pathway. And so tau is a protein that stabilizes microtubules. It's abundant in neurons of the central nervous system. It's a product of the splicing of the genes known as MAPT. And when it becomes excessive or hyperphosphorylated in the brain, it develops neurofibrillary tangles, which are also commonly associated with Alzheimer's disease. So we have 3 research questions. The first one is which APOE allele is most responsible for neurodegeneration in Alzheimer's? The second question is there a correlation between APOE alleles and tauopathy resulting in neurodegeneration? And the last question is what is the effect of APOE on astrocytes and neuronal integrity in mice compared to the absence of APOE? So some of our mouse models, so throughout our entire experiment, transgenic mice are used. So transgenic mice were created because mice also have an APOE gene, like us. So they only have one though, and there's the one is most likely represented by our E3. And the way the transgenic mice are created is by using recombinant DNA, and it's implanted into the mother. And so when the offspring are born, they're born with this transgenic gene or desired gene that was inserted into the mother. So for us, we used a mutant P301S of tau. So it's a form of tau that was inserted into the mother. So all of our offspring have this tau protein in them, or tau mutant form of tau. So the way the APOE was incorporated or the human APOE was through knock-in and another method of knockout. So knock-in is when they insert a desired gene into a specific locus. So and into the locus of a targeted genome using homologous recombination. So all of the mice after that were born also contain the APOE allele. And the knockout is where instead of incorporating the APOE allele, they inactivate it or knock it out. So it's basically not functioning the same way using an artificial form of DNA. And this helps us to test the function of genes, so you can see how it functions with or once it's activated or once it's not activated.
So my research question is which OPE allele most responsible for neurodegeneration in AD? I was looking at 9 months mice. The first one is wild type, and the rest is TE2, TE3, TE4, and the knockout mice. For the fundamental concepts is using immunohistochemistry, and that is this diagram right here. And because this is just the basic, so we have 3 methods that first stain with the, trying to find the allele and stain with the different region of the mouse brain. For volumetric analysis is looking into the piriformal cortex, the hippocampus, and the like lateral ventricle. The neuron layer is the hippocampus, and finally the thickness measure of the granulocyllator that's going to mention in the future slides. As you can see, that these are the mouse brains, and the temporal cortex is this outer layer. The hippocampus is the yeah. And
You can use mouse to help her too by using that.
as you can see, the wild type and the knockout brain, it look really similar. While the T2, T3, and the T4 is having degeneration, that you can see that the degeneration is correlated to the graphs. And I highlight the TE4 because it had the most degeneration of the cortex, the hippocampus, and the ventricle. It have bigger volume compared to these two. And in this figure, with figure C, show the granular cell layer. That's basically the ridges or folds in the brain. And as you can see, I highlight TE4 again, so how thin compared to the rest, while wild type and TEKO is almost the same. That correlates to the graph. This shows a strong positive correlation between APOE alleles to degeneration.
So the second experimental design or question is if there was a correlation between APOE alleles and the tauopathy regarding to neurodegeneration. So here we have the transgenic mice, and they were chosen at 3 months and then analyzed again at 9 months because we know neurodegeneration worsens as you get older. So the first thing that was done was brain extractions were taken from each of the transgenic mice from wild type T2, T3, and T4, and the knockout. So that was to because that's where you have the proteins and the tau is there, the APOE are there. So ELISA was performed, and this is an immunotechnique. It's an enzyme-linked immunoabsorbent assay. So this technique is used to detect and quantify proteins and antibodies. So it was used here to try to detect the tau proteins or tau protein markers to know that tauopathy was happening or occurring in these mice, along with the APOE. So the Western blot and the RT-qPCR tests were done to test the abundance of these proteins and to see to test also the level of neurodegeneration. And the phosphorylated tau staining was done to test or to see the visibility of the effect of the APOE along with the tauopathy on the brain extractions. So some results. So in the green box you can see here are the tau markers. So you have the RAB, RIPA, and FA. And the top 3 are for the 3 month, and the bottom 3 are for the 9 month. So here you can see, so just to kind of tell you what everything means. So the WT is obviously wild type. The T with the E2, T3, and E4 are the tau with the APOE allele, and the TEKO is our knockout. So here you can see that the TE4 does have the highest, neurodegeneration compared to the other alleles. And as you can see, only 2 of them are selected because those are the only 2 that are statistically different. As you can see at the top, there are p-values up there. So on the other 2, it might appear like TE4 is also a bit higher, but it's not statistically different. So you can't conclude that to say that TE4 was a part of or increasing the tauopathy. So another further data was done on the staining. So the type 1, 2, 3, and 4 were different types of staining. So see there's kind of a trend between the knockout and the E4. So for types 1 and 2, you see the knockout has higher levels or is more visible compared to the TE4. And the type 3 and type 4 were higher on the E4 than the knockout. So this kind of shows a trend and kind of correlates with the idea that the knockout and the TE4 have different effects. So here we obviously know that the E4 has a higher exacerbates the tau, while the knockout might be having a reversal or protects some of the pathology that's occurring along with the neurodegeneration.
Tristan, can I ask you to do as a favor? Can you go to the board and look
sorry, I
at
can do
explain
that.
those trend to us a little bit
Yeah,
more?
so these two here are for our knockout for type 1 and 2. You can see they're a lot higher, where here the T4 is low and there's almost like none for type 2. And then for the opposite here at the end, you have obviously a lower knockout for type 3 and type 4. Sorry, I'm totally bugging you guys. And then you can see it's also higher for the T4. So that's the trend we're talking about. And the two, E2 and E3 kind of are the same, which is kind of known because the APOE E2 and E3 kind of have a similar effect, where it's not as drastic to the E4.
So when you say higher or lower, who are you comparing with?
You mean like visibility of the screen?
Right, because you're saying TKO is higher compared to
Okay.
boom, right?
Sorry.
And then T4 is lower compared to boom.
Okay. So we're comparing that just to the TE4 is higher or lower. When I say higher or lower, I mean it's more visible in the staining compared to the E4. So I'm comparing the E4 to the knockout when I'm talking about this, not to the other ones. Because that's the trend that we see. That makes sense.
All right, thank you.
So for the third experimental design, we had a question of what is the effect of APOE4 on astrocytes and neuronal integrity in mice compared to the absence of APOE or APOE knockout. So for this, they took RNA from 3-month TE4 mice, 9-month TE4 mice, and 9-month TE knockout essentially. And they did reverse transcriptase, RT-qPCR, where they converted the RNA to DNA, to make cDNA, which then can be used for PCR, polymerase chain reaction. And then this was, I believe, analyzed using a dynamic array integrated fluid circuit, which was used to assess the microglial gene expression profiles associated with activation of A1. And according to the paper, a recent study was shown that A1 astrocyte inflammation-induced reactive microglia produce specific cytokines or inflammatory molecules that can activate A1 cells, A1 astrocytes, which then lose their normal functions and secrete toxic factors that can induce neuronal death. And so that's the first portion of the experiment. The second portion involved, I believe, glial fibrillary acid protein stain, or GFAP stain. GFAP serves as a biomarker for inflammation-induced astrocytes. They wanted to indirectly and qualitatively assess the number of astrocytes activated in each mouse model, particularly between the knockout and the TE4. So essentially, they followed a procedure where they did a fixation. They cut out a portion of from each mouse model. Did a fixation, dehydration, clearing, infiltration, and embedded the tissue section in paraffin wax. Then they use a microtome to slice different sections to be stained for GFAP, the protein. The third portion of the experiment involved Western blot of the GFAP protein, which is similar to the second one. In this case, they wanted to quantitatively assess the GFAP protein done using the amount of GFAP protein in mice model. So using the Western blot in this case, and with a primary antibody against the protein. And so that's the general procedure. They have extraction, then they do SDS page gel of the protein. They transfer the protein onto a membrane, then they use antibodies against it to be visualized. The fourth portion of the experiment involved a a co-culture of primary wild-type neurons. So these are neurons that weren't altered, weren't grown. They just take it out from each from the different mouse models, and then these were infected with human tau and mixed with glial cells derived from human APOE knock-ins, or TE2, T3, T4, and then you're knocked out. And they looked for they looked for the effect on a number of wild-type neurons co-cultured for each mice model. So for the first result, they saw that APOE4 leads to robust astrocytic activation, whereas the absence of APOE results in less astrocytic activation. So what they're primarily looking at is A1 specific, so A1 astrocytes. And this region, the red corresponds to more activation, and more blue corresponds to less activation. And according to the data, they found that there was significant activation of A1 astrocytes in 9-month TE4 mice, boxed in green here. And as you can see, here's the A1 section, the red corresponds to more activation. For the 9-month knockout, there was less activation of astrocytes. Interestingly, there was very little or no activation of A1 astrocytes in the 3-month TE4 mice. My guess was suggesting that since Alzheimer's is age dependent, they wouldn't expect to see that early on during the mice development. Okay, so other studies not only showed that APOE4 led to robust astrocytic activation, it also promotes neuronal death, whereas the absence of APOE results in less astrocytic activation and preserved neuronal integrity. So GFAP can serve as a biomarker for inflammation-induced astrocytic activation. And according to part B of this figure, especially here, this is the TE4, so E4 means the allele, and this is the knockout for the APOE. A brighter region, yellowish, orangish, indicates a higher signal of the GFAP protein, whereas darker regions shows less. So from here you can see that there's drastic elevation of GFAP signal observed in 9-month TE4 mice compared to knockout, the knockout version, which is indicated in blue. Comparatively speaking, you can also see that TE2, TE3 are similar, while the wild type has no signal, essentially. Part C of this figure is the quantitative version of part B, showing you again that there's higher levels of, by person area, of TE4, expression of GFAP in TE4 compared to the knockout, which is barely seen there. TE2, TE3, and the wild type is also shown there. Again, there was 2 regions, the hippocampus and the cortex were analyzed. For the cortex, the same thing, the same pattern is observed, where there's a higher level of GFAP for TE4 mice models than knockout. Part D is the amount of, they found for part D the amount of GFAP highly correlated with brain volume, where the greater the GFAP percent area, the smaller the brain volume. So, for example, here, the x-axis is the percent area of GFAP, and the y-axis is the volume. So as you have higher amount of GFAP, there's less brain tissue observed. Same thing, the correlation is 0.66, which is relatively high. Same thing is observed for the cortex as well. And E, part E of this figure just shows the Western blot results, again indicating that TE4 has a higher signal and much more GFAP expression than the other mice models. So the last portion of the experiment involved co-culturing wild-type primary neurons infected with human tau with a mixed glial cells derived from human knock-ins. E4 specifically led to the greatest neurodegeneration compared to the other knock-ins, E2, E3, and the knockout. Part F of the figure shows, the qualitative results show pronounced neuronal death was observed in E4 co-culture compared to other co-cultures. So the green essentially indicates neurons. It's very clear there's less here than the other the other mice models, especially for the knockout, has the highest retention of neurons. And part G essentially is a quantitative analysis showing that shows the lowest neuron number of an MAP2 percent area associated with E4 co-cultures, while the highest is associated with EKO. MAP is a microtubule associated protein 2, which is apparently important for mammalian brain development. So it's another way to kind of see whether what's leading to the neuron death. So with less of it, shown here, less of it is more associated with the the E4 mice model than the other mice models, especially the EKO, which has the highest. Highest TNF alpha cytokine, which is associated with systemic inflammation, was observed in E4 co-cultures, and the lowest was observed in the knockout model. So in conclusion, APOE4 has a detrimental effect, while the absence of APOE has a protective effect on the brain. APOE plays a key role in tau-mediated neurodegeneration. Specifically, APOE4 allele caused the most neuronal damage compared to APOE2 and APOE3. APOE knockout mice showed a neuronal death protection. APOE also plays a significant role in neuroinflammation-mediated brain atrophy. Degeneration of neurons induce neuroinflammation, which is most exacerbated by the APOE4. So here's a small schematic of like the hypothesis of what they believe led to brain atrophy. You have APOE, which is associated with neuronal death, specifically the E4 allele, the E4 version, whereas the knockout version had the least damage to the brain. This then induces brain atrophy, or you can have the associated neuronal death from E4 essentially causing neuroinflammation, which then can lead to brain atrophy. Or this can directly lead to tau pathology. Also, the APOE itself can directly possibly cause tau pathology, which then can lead to brain atrophy again.
The significance of it that scientists have gained more understanding of the effects of APOE alleles as they pertain to neural damage. APOE provide protection against neuroinflammation, and targeting APOE, especially APOE4, may be promising therapeutic approach in reducing tau-mediated neurodegeneration in the future.
So some limitations are that they haven't been tested on other animals. That Alzheimer's disease is a multifactorial disease, so it's hard to pinpoint the exact cause. And because of that, there's multiple hypotheses. So there's two other pathways or hypothesis pathways that have been presented. So they both start off the same, but towards the end is where they switch or have different ideas. So they have the APOE inducing tau levels or having different levels of tauopathy, and which leads to the different alleles having more effects. But once you get to the alleles is where they change. Whereas B, you see that they believe that the alleles cause neuron death or neuron damage, which leads to brain atrophy, which causes an immune response, which is the neuroinflammation. And then others also believe that from the alleles or the E4, you get neuroinflammation or triggers inflammation of the brain, which can cause that inflammation then